All right, Mike, you can get started.
Okay, well, welcome everyone to the Explorers Club Monday Night Lecture Series. This is a very popular series and is growing in popularity, and I'm very proud and happy to be here as your master of ceremonies tonight. And fortunately, I've got good content to present tonight, so that's a good thing. Before we get started, though, I'd like to thank a few people. First and foremost, Ann Passer, who is in charge of this program, has done a spectacular job with the wide variety and the excitement that's been caused by this whole series. Also, I'd like to thank Santiago Pardo Sanchez and Kevin Murphy as well, who have been very supportive from the technical aspects of getting these lectures up and running. So with that, I'd like to just give you a little background for me. Why am I here tonight? I'm a fellow of the Explorers Club. I'm a Sweeney medalist. I had a long history of exploration in the field with, particularly with expedition medicine. And I've been a medical director or leader on several remote expeditions, including going to the Titanic, Central African jungles, to the Gobi Desert, the Amazon, Antarctica, and I'm a consultant to the National Geographic Society. I'm also a contributing editor to the Explorers Journal, where you may have read my column on expedition medicine. And I've published books in these fields. So tonight, it's a great pleasure of mine to be able to talk and introduce and talk with Dr. Martin Nweeia, who's not only a personal friend, but as a scientist that I highly admire for the amount of work that he's done, particularly with the narwhal, which is a very unusual animal. And he's one of the foremost experts in the world on this. Particularly, what is the tusk? As you know, that's the only tusked whale. And he'll correct me if that's wrong, but I think that's correct. At any rate, it's a fascinating, anatomical part, and he is a major expert in that. And they've published widely on this and found a great deal about this, which he'll explain to you. But Martin's also a fellow of the Explorers Club, and he's carried the flag on 14 high Arctic expeditions, many of them to the area where he's going to discuss tonight. He received the Lowell Thomas Award for his Arctic research. He's a lecturer at the Harvard School of Dental Medicine and is an expert also in dentistry as it relates to work in remote areas. So he and I've had a long association regarding that particular aspect. In 2017, he was selected by the US Embassy as the Ambassador for Science at the opening of the International Pavilion in Ottawa to celebrate the 150th anniversary of Canada. Martin has received 9 research grants from the National Science Foundation, as a National Geographic Explorer and former research fellow in anthropology and vertebrate zoology at the Smithsonian. He's currently a research associate in the Marine Mammal Program at the Smithsonian Institute and in the Department of Vertebrate Zoology at the Canadian Museum of Nature. Dr. Nweeia's work has been featured in 4 documentaries from National Geographic, 2 from the BBC, and 1 from the French Découverte. So he's widely recognized for the amount of work that he's done up here, and I'm telling you, this is a fascinating project. I also have sent Eagle Scouts to him on a very special project up there in the Arctic, working with the narwhals. So without further ado, I'm going to turn this over to Martin because you'll be fascinated by his program. We've got some interesting discussion after his presentation, so stay tuned. We will have time for you to ask questions, and you can call them in or write them in as directed to Ann and Kevin, who will then relay them to us so that we can help answer them. So without any further introduction, I could go on for a while about Martin's qualities and characteristics, but I'd rather let him speak himself about the work he's done. So Martin, please join us, and I'm going to sit back and enjoy your talk.
Oh, good. I, like you, Mike, want to say thanks to the Explorers Club for hosting this series, and thanks to my gracious host, Mike Maniak, for kindly introducing me. This has been a fascinating year, as most of you know, and I have been able to conduct 2 expeditions this year with some difficulty, but I am pleased to say that everything was indeed successful. I'm going to start by sharing the screen here, and we'll get launched into what I hope will be fascinating for you. Let's start right here. Everyone see that okay? We're good? Let me go back just one. So this is about Narwhal Arctic expeditions and overcoming the restrictions of COVID. As most of you know, a lot of organizations like the National Science Foundation, National Geographic, Explorers Club, we all had to kind of limit our field of exploration this year. And I do my research in two zones that were regarded as green zones, one of the few areas on the planet where COVID was pretty much non-existent. Now in Nunavut, there are several cases, and Greenland has reported several cases, but for the most part, restricted travel to both of these areas has enabled them to maintain a non-COVID environment. And certainly this is very important in both of these environments, only because there's very little medical substructure that can really support a widespread pandemic if it ever hit that area. So I think the caution was certainly justifiable. When I first realized that our work would be canceled or postponed this year, I was trying to think of creative ways to see restriction as opportunity, which is what we as explorers do, right? I mean, all of us have been in situations where everything has gone wrong despite the best laid plans, and we have to think on our feet as to how we're going to overcome some of these restrictions and some of these issues. I always tell people there's no such thing as a problem, only a solution. So when I was first envisioning how to collect samples in Nunavut, since I was restricted to travel there, unless I wanted to undergo 14 days of quarantine, first in Canada, then another 14 days in Nunavut. So I would basically be in quarantine for almost a month before I would even be allowed to go in. And even then you require some diplomatic papers. So I enlisted the help of collaborators I have met and known along the way. And it speaks well to how we do collaborate in science and with the Inuit, who have been partners with me all along this journey. And I think it's a testament to how well you can form collaborations, because it gives you so many opportunities to overcome some of these kinds of problems. So I enlisted one agent up in Pond Inlet, where I was planning on collecting the samples, to review some of the protocols that we had done. We exchanged all this information, and he kindly agreed to collect samples, which we did. I sent up a dry shipper. I don't know how many of you have gone on expeditions with a dry shipper, but there's always some reticence, because as soon as you mention anything that has to do with liquid nitrogen, it doesn't make many people smile on air carriers. But fortunately, we were able to get the dry shipper successfully up to Pond Inlet, which in and of itself is something you're kind of on nerve about. And the samples were indeed successfully collected, had them shipped down to the Canadian Museum of Nature. And keep in mind, we still have to go through all the permitting process. So, and I have never had one instance where I collect samples where I have not personally been with them that has been successful. Something always inevitably goes wrong. So I was waiting for certain issues to happen, but fortunately enough, I've been through the experience so many times, I kind of knew the roadblocks, the obstacles, and we were indeed successful. So from there, those samples were sent to the frozen zoo. I don't know how many of you are aware of the frozen zoo in San Diego, but this is a genetic repository of all kinds of cell lines for animals, so that ongoing research in the fields that I'm now engaged in can be conducted. And so we are in the process of doing that now. Obviously, with limits in staffing and things, this will probably take a little bit longer of a process, but we will indeed be utilizing these cell lines for some of the research that we'll talk about. This next slide is Sedna. This is the goddess, the Inuit goddess of the sea. And as you can see, all her marine mammals in her hair. I've got the USAID logo up there only because they helped sponsor another expedition I did in Greenland, where I physically was there, but only got permission because there were diplomatic papers that were provided to me. And even then I had to undergo 10 flights, 4 COVID tests, and a quarantine. And I must tell you, it's a little bit more than I could predict because everything was kind of a moving target, if you will. I had later discovered that if one person on any of my Air Greenland flights tested positive, that we were all in mandatory quarantine for 14 days. So even when you're successful at arriving under these kinds of conditions, there's still considerable risk. Now, the first area, because Mike had alluded to some of the research that we are conducting currently, and there are three major areas and three major publications recently that I've been a part of. And the first one was the brainchild of Dr. Harris Lewin at the University of California at Davis, who decided to sequence the ACE2 receptors in 240 mammals that we had comprised genetically with the Broad Consortium, which is now called Zoonomia. So if any of you go online and Google Zoonomia, you will see the current research that has just been announced there. But this was a paper that we had done to look at the risk across mammal species for the SARS-CoV-2 virus. Naturally, a lot of attention has been placed on human risk and what has been involved, but certainly for the ACE2 receptors, which are involved for the most part in blood regulation, angiotensin-renin systems, for those of you with a medical background, there are roughly 25 binding sites that have been discovered with these receptors. And of those receptors, certainly in primates and in humans, we have 25 potential binding sites that can combine with the SARS-CoV-2 virus, with the spike proteins. And because of this, obviously there's considerable risk for people conducting research or visitation to areas where there are nearly threatened or threatened primate species like lowland gorillas, which obviously share 25 of the same 25 of these binding sites that we do. But interestingly enough, when we looked at this research, we also found that whales, like the narwhal, were actually in a high-risk category. We divided them into 5 risk categories, starting from very high, which were obviously humans and primates, then going down categorically. And whales, interestingly enough, many of them, especially toothed whales, were in this high-risk category. And that surprised many of us in the group, including obviously myself, because the beloved narwhal is sharing 22 of the 25 binding sites of this virus. So naturally, it has a propensity and a possibility to indeed bind this virus. Now, in terms of understanding what that real risk is, if you look at the background of coronaviruses in general, there already have been coronavirus infections in marine mammals. Certainly not one like the SARS-CoV-2 virus, but alpha and gamma coronaviruses, since there are categories of them, have been found in these species: bottlenose dolphin, beluga, seal, and other viruses have also spread. The distemper virus in 1988 and 2002 caused about 50,000 deaths amongst marine mammals. And keep in mind, these figures are all obviously very conservative because no one's out in the ocean monitoring marine mammal species for viral infections. So the unique part of this paper, though, and what I had mentioned to Harris Lewin at the time, is it represents a giant leap for a field of conservation genomics. Typically, when you go to lectures about conservation, everyone is talking about how to solve a problem. So something has already happened, and we're trying to recreate either an environment or a condition where a species that's threatened can have a better potential at surviving. Now we have the ability with conservation genomics, like this paper, to say, okay, look, these are species that have either high or medium risk of catching this virus. What are we going to do as a world society to either prevent this or to enable an environment where it's less likely to happen? And obviously in this situation, not to focus too much attention on a waste plant here, but waste is obviously one other pathway that hasn't been explored as much as the common one that's really referred to as aerosols. everyone really refers to aerosol, since we're all wearing masks these days, as the way to prevent viral spread. But certainly this virus can be spread with fecal material as well, and black water waste. So then we have to start looking at, well, how are we dispersing and how are we dispensing our waste? And I can tell you, and I always use this example because I'm from Connecticut right now, and we're talking from Connecticut. But we had a power outage here about a month and a half ago, and our main wastewater plant had a faulty generator, backup generator. So it wasn't working. So it allowed a little over 1 million gallons of untreated waste to go into the Connecticut River. Now that's in a progressive place. That's in an area where everything is working great, supposedly. Now we have to go to other places like Nunavut or Greenland, where I just returned from. Greenland, for example, has no wastewater treatment. So all of this is going into the ocean. And I think, you know, typically when the world of toxins occurs, people always say, well, dilute, dilute, dilute. Well, now that we have 7.8 billion people on the planet, it's pretty hard to dilute all our waste. And it's even hard to get rid of our waste. So it's piling up in many areas, and we're thinking of creative ways of obviously dispensing that. For any of you who saw the documentary, The Great Blue, that dealt with a little bit of some of the creative ways of dispensing waste, but certainly more creative ones have to come about. So the other publication that I've been a part of was just recently released. It's the cover of the most recent Nature journal. Again, I've been fortunate to be a part of the Nweeia. Oh, I see it's spelled wrong. Oh dear. So it's the Nweeia Consortium at the Broad Institute at MIT and Harvard. And here we have collected a lot of the brightest minds, I think, in genetics to really find the value in understanding multiple species across multiple lineages. So we can have a better understanding of comparative genomics and understanding how genes evolve, what kinds of traits are existing, why that occurs, and having a group that has already established so many of these genomes, in this case 240, you can create a lot more interesting data sets, obviously, with this many genomes. So we have been doing the same thing with the Narwhal Genome Initiative, which I have led for about the past 10, 15 years. And again, with the help of many collaborators, there are at least 50 to 100 collaborators in this. So I dare not start with names because I'll inevitably forget somebody. But I must say I've been gratified again by large collaborations. And in genetics, this is kind of the way forward. Everyone realizes in this field, it's so involved, it's so intense and labor-wise, so many steps that need to be done that you really need to rely on lots of people. And they're very comfortable collaboratively. So I have found this group immensely collaborative and very helpful, and have allowed us to really create at this point the reference genome for the narwhal. And indeed, we'll start asking some of the more interesting questions. In this case, on an earlier paper, we identified 4 genes that dealt with sensory function, since that was one of the main components that I had initiated in a 2014 article we'll talk about a little bit later on the function of the tusk. And this was the last publication that I've been a part of recently. So all of these have been out within, I would say, the last 2 months or so. So they're very recent. This is again a larger consortium. It's part of the Smithsonian Arctic Crashes project. I am lucky chapter 13. As my wife says, this is the goddess number. So I'm very happy being labeled with number 13 in this case. And what we're really talking about are some of the stability of species, because obviously everything is in flux in the Arctic. I mean, we all hear about change happening at 2 to 3 times the rate of the rest of the planet, but rarely do people go into some of the details. For example, caribou, which had a population 20 years ago of about 5 million, now down to 2 million. Large changes in environment, large changes in species. And again, how are we as a society going to deal with this? How are we going to navigate science as it does deal with it? I should say that in that Arctic Crashes chapter, mine specifically goes very heavily into Inuit traditional knowledge, showing the value of their categories, how they classify the narwhal. It's much more complicated, much more in depth than the scientific version. And again, without these partners, folks like myself in science really can't stand alone. So these people obviously stand with me even as I present. This is the Inuit calendar. It's based on 6 seasons. It's guided by many environmental factors that you can see, phases of the moon, sky, stars, ice, water, and species. And now that we have these 6 seasons, as we've discovered even with our 4 seasons, things have been a little bit mired in terms of distinct boundaries between what season you're in. So in the Arctic, for example, you could be supposedly in spring that could be very much like winter, or likewise you could be in a fall that could be very much like summer. With all of these unpredictable things, both in terms of not only temperature, but ice flow, for example. We all know about the ice sheet melt. We'll look at that a little bit later. But in terms of all these variables, this is an environment that is rapidly changing. Even ideas like magnetic north, which most of us think is a fixed area, is moving at the speed of 34 miles each year towards Siberia. So much so that the world magnetic standard had to be changed last year. So everything from your GPS to global navigation for ships all had to be changed because it's obviously related to this. So you can imagine the kinds of changes that are going on just with people and the species. Here, for example, I know we've all seen these kinds of maps, but shows the ice melt at the North Pole and some of the glacial melt. I'm going to let this animation run. Hopefully, I know animations sometimes work well and sometimes they don't on Zoom, but we'll give it a whirl. If it doesn't, it's okay. With all of these changes that are going on, we also have to keep in mind that in addition to changing chemistry in the Arctic Ocean, which already is a complicated ocean to begin with, keep in mind it has a subatlantic layer underneath it, warmer water underneath it, and those are separated by what's called a hydrocline layer, and which is a high salt layer. And that with all these changes that you see over time, this is having dramatic effects not only on the fresh water content of the Arctic, which obviously relates even to COVID transmission, right? Because many people use the argument, well, even if there's untreated waste, won't the salt water break down the virus pretty quickly? Keep in mind that during the summer months, the water in many of these inlets is very brackish, and there's tons of fresh water that are flowing in. And also keep in mind that if we were looking at COVID transmission between species, granted narwhal have a very restricted species, geographic area rather, but keep in mind beluga, which inhabit many of the cross territory that narwhal have, can also be found in estuaries. So theoretically, a beluga could have COVID and bring that up to the Arctic and spread that way. So it could either be from human to animal or from animal to animal. So there's many factors to think about. Also, the calcium, free calcium in the ocean is being bound as well during this whole process. And many people have talked about ecoskeletons and some of the lower echinoderms that are not being formed. But how does this translate to skeletons in mammals? You know, is this, because calcium is an essential building block for everything that's in the environment, including for us. But imagine the impacts that it has on the mammals in the Arctic. So here I am. This is last week, believe it or not. I am on top of what they refer to as the 660 glacier outside of Kangerlussuaq. I must tell you, you feel like you're on the moon here. And I was delighted to be there more closer to winter because I was told that the ice takes on a whole different sheen here. I am with flag 211. I had a flag that was sitting and I wasn't sure if it was going to be used, and we were allowed to take it on this expedition. So it had some right to at least make this trip, but fortunately everything worked out well. I must say for our audience, since we have, I'm sure, quite a few explorers, people that deal with ecotourism, this is not a pitch, but something that I have been involved with peripherally with the Agency for International Development in Greenland, because we're trying to allow opportunities in ecotourism to be siphoned to people that have this traditional knowledge, communities, Inuit, Inuit communities that have this tremendous knowledge that can be of assistance. So for any of you, even if you're conducting research, maybe you're leading an ecotourism group, maybe you're head of a cruise line, whatever your venture or aspect that may be along these lines of ecotourism, feel free to contact me. I'd be happy to put you in touch with people, because we're trying to create a larger nexus of people that are interested in sites and in websites. So there's a larger communication and bridging of knowledge that we can harness from the Inuit people in Greenland that can share with people who are visiting what will certainly become a larger visited area for ecotourism. These are some of the areas primarily for my research. As I mentioned, I'm primarily in the northwestern part of Baffin Island, northeastern part of Baffin Island as well, and also the northwestern part of Greenland. I haven't been to the eastern part yet, but will likely go there in the coming months. And this region that I work in is typically referred to as the High Arctic. It is very much aligned with communities that harvest narwhal legally. The Inuit people are the only people legally allowed to harvest this species. Obviously, it's an important component for nutrients in their diet, and it has significant social, environmental, and dietary impact for their societies. And this is the beautiful land of Greenland for those of you who've been through Disco Bay. A lot of the research I was conducting, this was back around the International Polar Year, around 2005 to 2007, when I had one of my first NSF grants. It was a 3-year grant, and we collected Inuit traditional knowledge from various communities, including Disco Island and Hunda Island, as well as Kanak and Umanak and quite a few other communities in northwestern Greenland. This is also out of Umanak. Again, I was not able to get to Umanak. I tried to get to about 4 different places. Air Greenland was actually changing flights even as I was there. So again, a lot of these aspects for travel now are moving targets, and certainly us and we as explorers understand how to adapt and just work with the changes. These are some of the folks, particularly from the Greenlandic side. Again, we had some from the Nunavut side as well, who were part of the research and the documentation. These are all found on the NSF site called Eloka, E-L-O-K-A. We had all of these translated, transcribed, and indeed, I am pleased to say we will have an upcoming exhibit as well that will display some of this knowledge and these people as well. They certainly deserve more attention. I was co-curator of the Smithsonian exhibit, Narwhal Revealing an Arctic Legend, as well as co-author on the William Mills award-winning book with the same title, if any of you want to get some further background. So here are some of the personalities and faces. This is Kajaningwa Kishut. I love this guy because, you know, I would like to say that he's smiling for the camera, but I must tell you, this is how he looks almost 24 hours a day. I wouldn't be surprised if he sleeps with this smile. He has since passed away. I've been trying to get information about him, but legendary because he would disappear into the wilderness alone, and everyone in his community, which was Kanak, which is a pretty hardy breed up there, always anticipated that something must have happened to him. Maybe he died, and then just miraculously, a couple of months later, he would just come in with the same smile on his face into the community. A loner, but a tremendous guy with a tremendous amount of knowledge. Each of these folks offered some insight. In this case, one of the things we talked about was the narwhal tusk material, which is quite unique in the sense that it is both flexible and hard in extreme. The most hard, the most flexible substance ever found on the planet. Certainly you can find harder materials and more flexible ones, but not in the combination that you find with narwhal. So it has unique biomimicry applications that we are currently investigating. Also, it has unusual flexibility in the sense that if any of you have seen a narwhal tusk, there's one behind me here. I won't grab too much at it so it doesn't fall. But if you've ever seen one, you've seen them hard and dehydrated. It doesn't look at all possible like these would bend and flex. And indeed, when elders told us that it could wave like a wand, I was there with the director of the Paffenberger Research Institute at NIST. We were both looking at each other like, is this possible? And believe it or not, the picture that you see on that lower left side shows the test that we conducted and indeed found that over a 9-foot section, the tusk could bend and flex 12 degrees in every direction. So I always tell people, you can almost bet 100% on one voice from the Inuit. Ole Quist from Umno, and colorful personalities as well. So we learn about the diet. So we were able to compose this slide because they're cutting open the stomachs of narwhal. And because narwhal swallow their prey whole, oftentimes they get their dinner for that night with a fresh fish that's sitting in the stomach. So at any rate, we can find out some of these dietary aspects, their feeding, their foraging, if you will, and different times of the year when they aggregate, all the behavioral questions. These are all composed with their knowledge. Richard Broberg, again, you know, each one is a story in and of itself. This is Jens Rossing, very legendary in Greenland. There's even a stamp that he's helped design. A very well-known artist as well as a wildlife investigator, did some of the anatomic recordings for the Inuit and how they classified narwhal that I helped use and integrate into the chapter that I did for the Smithsonian book. So very important. I went to his house, met him, and saw some of the samples he had personally because they're not available in any museum and they're quite unusual. So we're able as scientists to compose more accurate plates of anatomy. In this case, these were done from CT scans of samples that I had brought back, classic male, classic female, and a classic fetal skull. We also could learn about developmental patterns for the growth of this tooth. On the bottom, you see what's called these deciduous, or I should say, on a pathway of evolutionary obsolescence. They're accessory teeth, come in all kinds of unusual shapes and forms, but not considered fully developmental and likely on the path of being phased out. And I always tell people, keep in mind when you're looking at a trait like this over time, we don't know where we are on that evolutionary line of observing a species. So when we see something, people always ask me, well, if it's sensory, why don't females have it? Maybe they did have it. They certainly have it now. 15% of them do. And there are some males that don't have a tusk. So certainly its expression is curious and certainly is unprecedented in mammalian biology. I always tell people I'm reluctant to use superlatives in science, but in narwhal, you can feel very comfortable talking about their teeth with superlatives because there's literally nothing like this. This is Rasmus Avike from Qaanaaq. He was the one that shared to us about molting of narwhal. I had been with career scientists. We had talked about why narwhal migrate to these freshwater inlets. I remember the resounding term was, we know they're not molting. Everyone had observed beluga molting, but indeed after Erasmus told us about it, and it did with an open interview question. And I kind of stopped him and I said, could you ask him to repeat that? And indeed he commented on a story where he was kayaking next to a narwhal. And he said the outer layer just came off like a piece of gauze and immediately dissipated into the water. And then you can wonder why no one observed narwhal molting, but also shines a light again on these natural observers who are there for hundreds of years at times when science cannot be present, and harbor this kind of knowledge that I think, although we are understanding, they are being integrated, need to be taken on more fully as full partners. This is Pavia Nielsen. I supported him on an NSF grant to represent the Greenlandic Hunters at the Inuit Circumpolar Conference back around 2006, since they had reported many more narwhals than some of the scientific investigators. And again, this is a collaborative effort that should be hand in hand between hunters and science, as often it is, but certainly there are conflicts that occur as well. And we just want to make sure that these folks are represented in international forums because they're not being viewed in publications. Naturally, in mine, they are full authors. So almost all of my publications have some Inuit mentioned somewhere, since I consider them full collaborators. Again, Jens Jeremiasen, who taught us that during climate change back between 2005 and 2007, narwhal changed their migrating pattern. So they used to go into Disco Bay going up and going down. Now going down, they go just straight down. They don't go in the bay anymore. So not only do we learn about these different populations of narwhal migration patterns, certainly science can stick a monitor on a whale to find out where it's migrating, but we can also harbor the knowledge again of these Inuit hunters who are observing them daily. And as a result of that, we were able to compose a map of migration. This was published in two journals, one at the Smithsonian Arctic Crashes book that I mentioned, and in another in a book about Greenland, which was also a Smithsonian book. So this again is roughly about a week and a half ago. The gentleman dead center with the glasses on is David Brown, who was my host from the U.S. International Agency for International Development. And we were meeting with the Hunters organization and Fishermen organization in Nuuk in Greenland. There we're showing part of an exhibit and some photographs that I had brought with me to share. And interestingly enough, when I mentioned the molting story in this very forum, 3 of the other hunters present also agreed that they had heard about this as well or had seen it. So we're constantly gaining knowledge. And the criticism, obviously, of science is what's your sample size, right? And typically, if it's one hunter that says it, science is going to retort by saying, well, how reproducible, how trustworthy? And I always say, when your life depends on an observation, it's probably pretty trustworthy. But obviously, we need a bigger sample size. So having 3 additional hunters, great information to add to our repository of information. I was giving 2 talks, one at the University of Greenland and one for the Institute of Natural Resources. Both were widely attended, and we had great input from the students, very engaged folks. Was a pleasure to be able to show them the integration. This is a photo we did at the end of the talk. Obviously, we have to have a little fun. So we're trying to create the next batch of community members that can feel an alliance, a link, if you will, to science, get them interested in this. This has been a high priority for multiple national science organizations, including our National Science Foundation, as to how to get Indigenous peoples involved in science. And I always tell them, well, I mean, why would they? You know, they think differently, they respond differently, and we have to find a way to reach out to them and honor their knowledge, first of all, and give it credit and due credit, but also understand those differences and see them as both positive agents, which they are. Done collaboratively, they bring the best of both worlds, if you will. So again, this is the whale that kind of captivated my attention. You're probably wondering, why would a dentist from northwestern Connecticut travel 1,500 miles straight north to be in frigid water, unpredictable weather? I've been in an Arctic hurricane. I've seen probably more things than I would care to summarize. But having gone backwards, if someone told me all I would be going through, I'm not so sure how readily I would have signed up. But being on a pathway to do this, it also makes the knowledge more valuable. This was some of the studies we showed when I was talking about the tusk being built in a reversible manner, that it's built actually as a steel core in the center and very flexible on its outer surface, which allows this bending, the 12-degree bending that we talked about. And also this lineage. If we look at toothed whales, clearly there's one that doesn't belong in the group. And that's why I, as a dentist, was curious. Nothing about it really made any sense on any level, including the explanation for why it even existed. And secondary sexual characteristic, by the way, is a very convenient term. But keep in mind that even Charles Darwin, who categorized some of these animals, even he scratched his head. He didn't sleep very well at night with some of these examples. So this wasn't done out of understanding as much as it was done out of convenience, since the understanding of what a secondary sexual characteristic obviously goes a different level and a different depth. I always tell people that your understanding of anything is directly proportional to your curiosity. You can stop wherever you want, but you can keep going too. This is the narwhal mouth, not a tooth in it. Again, some of the Inuit work that we had been doing with some of the hunters about some of the migration patterns. And another factor that the Inuit had told us about, the savacit. These are ice entrapments of narwhal. More of them have been found over the past 10 years or so, again, related to fast ice formation, climate change as a byproduct to have this unpredictable weather, which entraps whales. And this primarily is an oral knowledge, but we were able to get some of the notes of some of these hunters. So I currently have another project underway, translating these journals into a book, because as I mentioned to the people involved in the project, I want the real investigators of narwhal to have their definitive book. So this will very much highlight their work, their understanding, and they as, in a sense, the presenters. This is the geographical distribution. As we mentioned, narwhal, very restricted species. Certainly there are other marine mammals with restricted areas like this as well, but amongst the Arctic marine mammals, and certainly the cetaceans, this is by far the most restricted species. And curiously, there's no analogous species in the South Pole that has the same restriction. The kind of work that I was doing, working with heart and physiologic monitoring of whales that had tusks that were being introduced to high salt and freshwater gradients. This was some of the older work that I had done, just to review quickly. Up above, again, you'll see a tusk with its nerve taken out at its full length, because very few of you would imagine seeing a narwhal tusk that is what's inside. But indeed, it's a full nerve, usually goes to the tip. Even if you cut an inch off of that tip, it would start to bleed. These were the tubules that we discovered in the landmark finding that we had in 2014, showing this to be a sensory organ. This was the heartbeat of the narwhal, first diagram like this by myself. Happy to be noted as the person who brought the narwhal heartbeat to a graph. And this was the mechanism that we utilized to show that indeed it was a sensory organ. That between this nerve and its outside saltwater environment, there was a unique pathway for these tubules to help this whale understand its external environment. Interestingly enough, blood plasma, the interstitial fluid in these tubules, and the ocean are almost chemically identical. There's very little differences between all three of these. This was the book I mentioned earlier, because we're now in a pathway of trying to educate people, how we get the next generation of scientists, but also the next generation of Inuit and Inuit and different Inuit populations circumpolar to be involved in science, to see it as something that's for them. And we're doing it on all levels. So both my wife and I came out with this graphic novel recently. It's available at multiple places. It'll be a signature book for the Smithsonian's traveling exhibit. We were selected by the Smithsonian for the exhibit to travel, obviously because of COVID. It's boxed up for a while, but this is another way to engage another generation of people so that they understand science, but it also integrates traditional knowledge. So there's a legend of the unicorn and a legend of the narwhal, in this case, that's told by the Inuit people, and that legend is indeed graphically represented in our graphic novel. So we incorporate traditional knowledge all through the graphic novel so that kids can understand how their knowledge is valuable to the process. The last thing I'm going to talk about is a project I've been working on with Hololens. This is from a product from Microsoft, basically wearing a computer on your head. It's augmented reality. And unlike virtual reality, which walls off your environment, you can interact with a group of people that are undergoing an experience. And under another National Science Foundation grant, I worked with Case Western Reserve University, where I am also an assistant professor in their dental school. And they are the proprietors of kind of the academic introduction of this into what's called Anatomy HoloLens. If you Google Case Western and HoloLens, which is spelled HOLOLENS, you will see a video that will completely blow you away. We did, these are all graphics from the experience itself. So this is a hologram that actually appears before a group of people. And it's huge, this globe. It's not just a small thing obviously depicted here. These are some of the graphic representations that are in the experience itself. We were kindly gracious enough to have Meryl Streep narrate the experience. So we were very excited. It was introduced at AAAS and the Smithsonian, and it'll be introduced in Greenland shortly as well. Showing again some of the experiences it directly relates to what you as someone in the experience see. By the way, you're in the ocean with the whales. You are physically there. And this is how it is for people in the experience. So these are two people in the experience with a whale, narwhal coming right at them. And again, the experience with people in the experience itself. And we devised a game at the end where you become a narwhal, can stun your own fish, since that was one thing that was brought up recently in research from the Canadians about this stunning that narwhal had the ability to do. So we did it as a game. And I wanted to end with this photograph by one of our fellow explorers, Gretchen Freund. For those of you who know her, a very accomplished photographer, as well as a very generous person. And I've always identified this photo as a significant metaphor for not only my work, but the work that we need to engage in the future. And that is as a collaborative effort. We're both out there, both Inuit science, working together at every step, including the publication step. And with that, I think we'll end and see if there are any questions.
Great, Martin. That was fantastic. I'm sure the audience feels the same way I do. And I've heard a lot of this story before, but it's fascinating to hear it again. And I know that you have much more that you could talk about. So unfortunately, we have to cut it a little bit short here. But your comments and your presentation raised some questions. And fortunately, we've had some responses from the audience. And we have a few things we want to go through with you. So we'll start into that. Going back to the COVID situation. So, I think the question that people might have is, what are the implications of this research for these endangered species, such as the narwhal? Should we avoid contact with these endangered species until this coronavirus outbreak is contained? Or perhaps should all who do research in this environment be vaccinated before embarking on the research? What do you think about that?
I do think we have to use discretion and caution only because we're still on a pathway of learning and understanding this virus and its transmission modes. Keep in mind, mink, for example, culled both in Denmark, the Netherlands, and Spain in the millions. This wasn't, this is in kind of our medium range for binding. And originally when it came out, people thought, oh, this was a human to animal transmission. And now they've discovered that it can go the other way. It can go from animal to human. So these were unpredictable. Same thing with the Bronx Zoo tiger, right? That has a low propensity to catch this virus. Why that? And because these animals with medium to low propensity for binding have obviously shown the ability to do this, we have to be highly cautious with endangered species and primates. So, anyone who's working with an endangered species, if it's on a reserve or whatever, I'm sure is using utmost caution, because it's pretty hard to teach a gorilla social distancing, so it could be catastrophic for a species. Certainly with narwhal, it's an active field that I'm interested in, just because I don't think there's really great ways of monitoring risk, but we have to understand our part and our job as a society in terms of, specifically waste disposal. So I hope that answers the question.
Yeah, no, that waste disposal issue is a very interesting one. I know there are other researchers working on that particular aspect. In fact, tracking waste by various means.
And certainly there are likely going to be research protocols where people have to test, maybe a PCR test, rapid test, whatever they want, and then quarantine before they can be, exposed to these other populations. We just have to exercise caution. And like I said, we have the ability now with conservation genomics to prevent a problem, not to chase after it and say, oh, now what do we do?
Well, we're going to go on to some other questions, but I do want to follow up with one question that struck me reading your paper, which was a great paper, by the way, great manuscript. And that is that, since the SARS-CoV-2 supposedly has come from bats, something that struck me very much there was the fact that the bats that were tested have either a low or very low binding score for the ACE2 receptor. Now, to me, that suggests that there's probably another mechanism for the virus in that particular animal species. I don't know. Do you have any thoughts about that?
Likely so, but again, it speaks to the issue of here we've identified a primary pathway of engaging this virus, which in this transmission species has a low propensity to do that, right? So it's a little bit counterintuitive.
Yes.
And in the same way, the story with the mink and the Bronx Zoo tiger and other cats, by the way, in other zoos have now tested positive as well. So I think it speaks to the aspect of caution, that there's a lot that we don't really understand, and until the full picture comes, and that's why science is around, right? So we can fill in some of this puzzle so we have a better understanding of how we, can be better stewards of our planet.
Well, that's true, and I think that's the big take-home message from all this is that we don't know very much yet on some of these aspects, and we're just learning about the receptors and what's required. There are other receptors, by the way, that are required for the virus to enter the human cell, and they don't talk much about those, but there's a serious amount of research going on in that direction. I'm going to move on to some other kinds of questions here because we've got a lot of people starting to chime in from the field, and let me see, will I read these? One quick question about the tusk. This is from Rhoda Septalisi in Washington, D.C., our Romanian princess on site there. And she wants to know, what can you maybe explain briefly, what's the difference between this type of tusk in the narwhal and tusks in other animals such as elephants?
Yeah, it's a great question. We're going to learn certainly more about that with gene evolution and an understanding of how, first of all, tusks even evolved. Because naturally, a tusk, although it's considered a tooth, has many individual properties that are not like teeth. Keep in mind at the base of a tusk, it's open, and the base of our teeth are typically closed with the exception of a nerve and tiny blood vessel at the base going in. So there are many aspects of that, but if you compare narwhal, let's say with elephant or walrus, keep in mind, narwhal is the only straight tusk. It's the only spiraled tusk. It's the tusk that exhibits itself in terms of dimorphism the most extreme. You have examples of male and female that couldn't be more different. It's the most diverse in terms of asymmetry. Imagine a 9-foot tusk on one side and a 1.5 foot tusk embedded on your right side. There are no examples of this in nature for teeth, none, zip. Again, you can, and these are just 5 examples quickly. I could go through probably about 30, but I'll limit it to this just because we want to get to other questions as well.
Yeah, okay, good. No, it's fascinating. I've heard you talk about this before, and it's just a fascinating organ, that's for sure. One quick question is that the movement of the change in the movement or the change in the position of the North Pole, why is that occurring? And is that related to anything related to do with the climate, or is this a cyclical process of some sort?
So this is magnetic north as opposed to geographic North Pole. So
right.
Many people may not realize, but over the course of our history as a planet, the poles have changed at least 3 or 4 times. So North Pole has become South Pole has become North Pole. So the whole magnetic field has indeed reversed itself. Are we in the midst of something like that? Obviously, this is something we're just visualizing more recently. So until, again, we have a better understanding of it, but certainly this has broad implications for lots of species. You know, you're talking about electromagnetic fields, which, you know, really have a lot to do with many features, perhaps of migration, behavior. You know, you have to think about why are some of these restricted species like that? Why do migration patterns happen in certain ways? And electromagnetic forces come into play in a lot of scientific investigations that deal with these kinds of variables. So it's an important one for us to consider.
Yeah, thank you. We have, I just want to point out that we actually have somebody from Nunavut who's tuned in tonight and is very happy to be here to listen to your presentation. So we're getting them from all over the place.
That's right.
Yes, it is. I think so too.
You can say Hanepe.
Okay, easy for you to say. Now, you know, the question that's parallel to some of the stuff we've been talking about is that given the proximity of marine animals to ship traffic, what are your suggestions for translating sewage management into international maritime law? Can the COVID virus survive in marine species, adopt and continue to be transmitted through the food chain to humans past the pandemic? Well, I'll answer part of that is that we know these RNA viruses mutate all the time. So yes, that's certainly possible. Things can change. But the first part of this question is interesting is, are you know, the sewage management from ship traffic, and how to?
We had mentioned this in the publication that I had spoken about, the first one in the Proceedings for the National Academy. You know, I've done considerable reading, and I'm sure there are lots of variables I may or may not be even aware of, but it has become apparent that even in vessels that may have treatment facilities on board, it has been questioned as to how well these are working. There's no monitoring of these systems, and indeed for other ships that may not even have a waste treatment facility. And this is in addition now to increased Arctic traffic. Obviously, because of ice melt, there's a higher propensity for cruise vessels to be going up the Northwest Passage. There's all sorts of research vessels that are engaged in these areas. The amount of boat traffic has increased quite a bit. Even in the brief 20 years that I've been up in the Arctic, you know, it has vastly increased.
Well,
so it's the same issue that I spoke to before. Some of these issues may not have been really problematic because it wasn't happening so much, or there weren't so many people, but when you look at the population of the planet, when you look at the increased volume of people now going to these areas for exploration, for research, even for a vacation, that we now have to be aware of how we manage things like waste. And I think there's ways we can improve this.
Well, and there's a parallel question that came up from the audience from Will Allen, actually, who wants to know if this increase in the traffic that you're talking about up there has had an effect on the narwhals.
Well, another one of our great explorers is Chris Clark, the former director of Cornell's Bioacoustic Lab. And I remember if any of you in the audience, have seen Chris's presentation, it's really stunning, because what you realize is that the same aspect of our sight being our main sense to understand our world, our marine life's ears and hearing ability to understand their environment. And when you consider all the background traffic from shipping lanes, from cruise line traffic, it's just a barrage of sound everywhere. How much are we interfering with, behavior, with song patterns of whales that are trying to communicate, and they have to now do this over an engine. Does everyone have to speak louder in the ocean to get their message across, and likely that's the case. And then you quantify that with the recent introduction, and not that everyone didn't know it before, but of plastics that have now gone to the Arctic Ocean. Now layer that on waste and how we dispense all these things, we are creating an amazing environmental soup that we really don't know the full ramifications of. And in this way, I think the Inuit, for example, could be our monitors. We should be enlisting some of these folks as ways, and I'm sure in governments they are, and they certainly communicate enough with each other that this is certainly happening, but hopefully it'll happen even on a formalized level so that, again, we can prevent problems instead of chase them.
A related question to the sound issue that you brought up. Do narwhals make their own specific sound? And has anybody studied any of that?
Oh yeah, lots of people have studied this. Bertil Moll was one of the kind of founding people that did a lot of work in this area, but and Chris Clark certainly has done work. There's been a lot of people that have done signatures for narwhal. I would also say that in terms of vocal range with cetaceans, narwhal has one of the more extensive vocal ranges. So we are just beginning to understand the significance of deciphering that language, since it happens on spectrum that we can only, we can barely record in some instances because it takes so much energy to record some of these sounds. But yes, there's a whole elaborate language and way of understanding behavior, migration, distribution, all of these things that sound plays a critical role in understanding.
I think we have time for maybe about 2 more quick questions here. So, and Kevin Murphy, you can correct me if we have more time, that would be great. One question relates to, comes from the leader of an all-female research team up there, Susan Eaton. And she was,
hello, Susan.
Okay, yeah, I thought you guys might know each other.
Yeah.
At any rate, she wants to know if you work with any female Inuit leaders.
Absolutely.
Yeah, that's what I thought that was true.
Yeah, I should have showed. I'll make a point to include her next time. That's as young people would say, that's my bad. But Elizipi'i O'Tuva, a name you might recall, has won the Order of Canada, was also one of the founding organizers for the Inuktitut to English dictionary, has been one of the primary people we have used to collect knowledge. And do I think more could be done with collecting the knowledge from women, absolutely yes. This is something, again, my wife is more interested in doing with Indigenous people around the world, to allow women to have their voice. Certainly within most of the communities that I was working with in regard to narwhal traditional knowledge, that men were referred to me by the community. How that decision was made, I don't understand the full way, but I do know that we got to some of the main purveyors of knowledge within their culture that were regarded as good information sources for the narwhal.
Okay, good. Thanks for replying to that. And another note here, Lou Tolman, who's in the Washington chapter also, asks if you have any contact with any of Matthew Henson's descendants in Greenland.
No, I don't. That's obviously a very interesting story. And I haven't, but the short answer is no, but I'm sure I peripherally have met them without probably knowing it.
Okay. And I'm sure Lou can contact you to discuss that further if he likes.
Sure.
So, yeah. Let's see, what else? There are a lot of questions coming in, so I apologize that we're probably not going to get to all of these. But there was another one in here that going back to what more needs to be done to get traditional peoples involved with research? There's some obstacles to that, I know, but
yeah, it's a great question, and it's one that at this particular time I am a bit consumed by, because I think it's a very important issue that has to be addressed culturally from their side and culturally from our side. From our side, I always tell people I didn't really learn history till I got out of school. That's when you get the real stories coming up.
Yeah.
And unfortunately, Indigenous people all over the world don't really have their story well told in a lot of history books, especially mainstream books, history books, within a typical elementary or high school. I think the same is true with science. You shouldn't have to get a PhD to start learning about how traditional knowledge can integrate with science. And I can tell you, as a science major, and having gone through 2 doctorates, I really didn't get my introduction to traditional knowledge until I started working at the Smithsonian. And you shouldn't have to wait till then to have that happen, which is one of the reasons why we introduced the graphic novel, so we can get kids at an early age to see how these can work together. That's where you have to start. You have to start planting seeds early. You can't do this at a late stage in development. So I'm currently in the process of working with several people. Harris Lewin, Dr. Lewin, who I mentioned earlier,
yep.
Club member Joe Rohde, who's been involved in our project as well as a few other people. We've had people from the Polar Trek program at NSF that are going to be involved, Kathy Ho. And the idea is to really address some of the significant differences between how these two knowledge frames interact, because that's how they have to be seen. I'm not going to go into it in depth, but briefly, science is very reductionist. We're looking for the one variable, we want to test it 100 times, and then we want to publish it. In the Inuit world, it's very, or Indigenous, for example, but certainly in the Inuit world, it's very contextual. They can't, it doesn't make sense to them to look at a variable. It makes sense. So when they describe a behavior, for example, like let's say a broken tusk or a narwhal tusking, they're liable to say, well, the weather was like this, the ice came in like this, I was with my brother, everything is contextual. And then they'll tell you how they saw it. They'll say, and I only saw it once, or, it's very much the whole story. They can't work the way we work. And And that's part of the difficulty in how they communicate and how they integrate. So the real challenge is, and there are lots of other differences too, but that's a main significant one that we will address in this, and hopefully come up with a product that is an educational piece for science textbooks. So we want to be integrated as a way for kids to learn this stuff at an earlier age, so people like me don't have to learn about Christopher Columbus, after we celebrate his holiday. I'd rather know the story before. And all those kinds of things make us a more interesting culture. The age of imperialism is over. People are tired of the same old format of people ruling other people and grabbing resources. People want different models of how societies work. And we want to be there with the educational piece that can lift that.
Great. Well, that's a good answer, and it's a complicated answer. And I think this is probably a good place to stop. I know there are several other questions that people have sent in, but we're just really running out of time here. So first of all, I want to thank everyone that tuned in tonight from a wide variety of places throughout the world. I hope you enjoyed the presentation. I certainly did. And the next time Martin and I are together, I'm sure he's going to tell me some more stories. So, which will be soon, I'm sure. And again, I want to thank Ann Passer and Kevin Murphy for all of the help in putting this program together. So next week's program will be on the next program will be on December 14th. And that's entitled Dragonfly, In Situ Exploration of Saturn's Moon Titan, an Organic Ocean World, and this will be given by Dr. Elizabeth Turtle. She's the chief investigator at Johns Hopkins Applied Physics Lab for the Dragonfly mission. So it should be a fascinating topic as well. And once again, thank you all for joining us. And Martin, thanks so much again for sharing your encyclopedic knowledge about this subject.
Yeah, thanks everybody for joining us. It's been great.
Goodbye now.
Yeah, cheers everybody.